Good morning, everyone. You guys can hear me?
Good morning, sir.
Hi.
Okay, so I think I'll start. Okay. So till now, last two class we discussed about the lipid chemistry part. Now from today, the we are going to discuss different metabolism which comes under lipid. So first we are going to see fatty acid synthesis. So slide is visible to everyone.
Yes, sir.
Okay.
Yes, sir.
Okay, so before we go on to the pathway of biosynthesis of fatty acid, you have to know a little bit background about it. So, biosynthesis of fatty acid happens in cytosol, basically cytoplasm part. We know that some of the metabolic pathways happen in mitochondria, some in cytoplasm. That is the major places in the inside the cell where metabolism happens. So, fatty acid synthesis happens in cytosol. In place of that, fatty acid oxidation happens in mitochondria. Now, for fatty acid synthesis, the most important substrate from which the fatty acid synthesis actually happens is acetyl coenzyme A. Because there are different types of fatty acids, different chain length, saturated, unsaturated, we cannot read about the biosynthesis of all. One of the most common fatty acid which is found in body is palmitic acid. So we are going to see the synthesis of palmitic acid from acetyl coenzyme A. But for all the fatty acid synthesis, basically, especially even chain fatty acid, acetyl coenzyme A is required. Short chain fatty propyl coenzyme A is also substrate. Remember that. So from where this fatty acid ultimately will come? So basically glucose, and we will see the sources of acetyl coenzyme later. So glucose is the primary substrate. So that's why when you take more and more carbohydrate also, ultimately whatever will get utilized, and some of the glucose will get stored as glycogen, and then rest of the glucose will get converted to fatty acid. So it forms fat lipid. So that's why excess of carbohydrate also is not good, because ultimately that converts to fat and gets deposited in the body. Now in type 1 diabetes, what happens, because insulin is low and insulin is required for the biosynthesis of fatty acid. Basically, insulin promotes fatty acid synthesis. So in type 1 diabetes, this biosynthesis of fatty acid will be reduced, and this fatty acid biosynthesis happens in many different organs. So important organ which you have to know is liver, then adipose tissue, mammary gland, kidney. Other important are kidney, brain, and lung. So most important is liver and adipose tissue. What are the cofactors which will be required for this pathway? So the cofactors are NADPH. We have to know from where NADPH is coming. ATP, by now you must be knowing, because carbohydrate metabolism is over. Manganese, biotin, and bicarbonate. We will see where all these are being utilized. So in the first step, remember I told acetyl coenzyme A is required. So acetyl coenzyme A gets converted to mannoline coenzyme A. That is the first step of fatty acid biosynthesis. And this first step is the controlling step, rate limiting step. Okay. And the enzyme required for this first step is acetyl coenzyme A carboxylase. So you have to remember the name of this enzyme, acetyl coenzyme A carboxylase. And in this enzyme, basically there is a complex of enzyme with biotin. We will see how this happens. The reaction steps takes place in two, basically there are two steps. First is the carboxylation of biotin, and then this carboxyl group which has been attached to the biotin combines with acetyl coenzyme A and forms malonyl coenzyme A. So if we see this, you will understand because it's easy. So this enzyme basically is acetyl coenzyme carboxylase is combined with biotin, and with the help of ATP and bicarbonate, it gets converted to enzyme biotin carboxylation. Basically, the carboxylation of biotin enzyme complex is happening with the help of ATP, and this enzyme biotin carboxylase basically gives its carboxyl group to the acetyl coenzyme A and forms manonyl coenzyme A. So for to understand this cycle, basically this reaction you start from here. Acetyl coenzyme carboxylase biotin complex, ATP and bicarbonate comes, reaction happens, it forms carboxylated enzyme biotin complex, and this carboxyl group of the enzyme biotin complex combine with acetyl coenzyme A and form manonyl coenzyme A, and again regeneration of enzyme biotin complex happen. So these things are written in your book. If you see this 2-3 times, you will be able to understand. For your purpose, even if you don't know the structure, it's completely fine. You have to remember the names, okay, of acetyl coenzyme A, malonyl coenzyme A. Acetyl coenzyme A contains two carbon, right? Malonyl coenzyme A contains 1, 2, 3, 3 carbon, and this is coenzyme A. Now that was the first step. Once malonyl coenzyme A forms, then the subsequent steps of the fatty acid biosynthesis will happen. For that, there is one enzyme complex which is involved. That enzyme complex is actually containing 7 different types of enzyme. We will see the structure of that, then you will understand. So this is known as fatty acid synthase complex. So first enzyme was acetyl coenzyme carboxylase. That was the first important enzyme of the fatty acid biosynthesis pathway. Next enzyme is fatty acid synthase complex. So this is actually not one enzyme. This is a combination of 7 different enzymes. We will read the name of all the 7 different enzymes. You see. I'll show you that first. Okay. So this is 7 enzyme. 1, 2, 3, 4, 5, 6, 7. Okay. So these 7 enzymes, 1, 2, 3, 4, 5, 6, and this is the 7th enzyme. Okay. And this is the carrier protein basically, acyl carrier of the fatty acid. It carries the fatty acid. So now these 7 are actually 7 different enzymes, but they are synthesized together basically. Okay. So functionally it works together, but actually these are formed from the 7 different polypeptide chains. So there is no barrier, diffusion barrier, but they are functionally separate. Okay, so when you read the molecular biology, we will discuss about translation, transcription, and all those. Na? So these enzymes are transcribed together, basically. That much you understand. And in human body, this enzyme remains in dimer form. First thing, this is a complex of 7 enzymes, and that 2 remains in dimer form. So 2 enzymes like this are attached with the sulfhydryl group and combines. Okay. So single polypeptide is 7 different enzyme activities in mammal. Acyl carrier protein is there along with that 7 enzyme and acyl carrier protein ACP. In book structure will be there. You have to go and see this again and again. So here the compartmentalization happens for different reaction steps, but the permeability barrier is not there. So when suppose one first step, just I am telling you example, ki first step is being catalyzed by this enzyme, so the product of this can easily go and combine with this enzyme, and then the product of this can easily go and combine with that. So diffusion barrier is not there, still the compartmentalization is there. Coordinated synthesis. So from one gene only these 7 enzymes are forming, and in mammal this complex also remain in dimer form. So this you have to go and see again and again. You have to remember the name: ketoacyl synthase, acetyl transacylase, malonyl transacylase. So if you notice, there are 2 transacylase: acetyl transacylase, malonyl transacylase. Okay. Then synthase, ketoacyl synthase. Then there are 2 reductase: enoyl reductase, ketoacyl reductase. There is one hydratase and thioesterase. You will see the involvement of these enzymes in the reaction steps next. So what happens first? Acetyl coenzyme A, remember whatever I am telling, those words you have to remember. Acetyl coenzyme A combines with that SH group. I will show you the enzyme again. Cysteine SH group of ketoacyl synthase. Okay, ketoacyl synthase and acetyl transacylase. This ketoacyl synthase and acetyl transacylase. Here actually this SH group is there and cysteine is there. So here the acetyl coenzyme A will come and combine. Okay. Then malonyl coenzyme A has formed, right? Malonyl coenzyme A has formed in the first step. It combines with the SH group of ACP malonyl transacylase. This is malonyl transacylase and this is ACP, right? So this the malonyl coenzyme A combines malonyl transacylase. That you can remember because malonyl coenzyme A is combining. With that you have to remember ACP. Okay, and for acetyl coenzyme A, it combines the acetyl transacylase and ketoacyl synthase. Then ultimately it forms acetyl malonyl enzyme. We will see next. Then acetyl group, it attacks the methylene group of malonyl residue and with the help of ketoacyl synthase. And this ketoacyl group undergoes 3 reaction steps: reduction, dehydration, and reduction again. So don't go after the structure. I will tell you the names, and you have to remember the names of the substrate, product, and enzyme. So this is fatty acid synthase multi-enzyme complex. Okay, just imagine. I told you this is dimer, right? This here the whole enzyme is there, fatty acid synthase. So it is dimer 1, 2. Together they are combined. So here on this one acetyl coenzyme A will come. Okay, and with the help of acetyl transacylase, it will combine with the SH group of ketoacyl synthase. Here you see ketoacyl synthase. With the help of acetyl transacylase, it will combine with the SH group of ketoacyl synthase. And in the first step, what has happened? This acetyl coenzyme A is also forming malonyl coenzyme A, and this malonyl coenzyme A, with the help of malonyl transacylase, will combine with the SH group of, it is coming here, right? SH group of the ACP, acetyl carrier protein, right? And then ultimately this, on this enzyme complex itself, acetyl coenzyme A, malonyl coenzyme A combining, and it is forming acyl malonyl enzyme. One thing I'll tell you to understand, you have carbohydrate metabolism over, so there you must have been reading that one substrate is there, enzyme comes, it forms product. On the product, no longer that substrate is attached or that enzyme is attached. Correct, right? But here what happens? The full synthesis of the fatty acid happens on this fatty acid synthase complex. So after the full synthesis of the fatty acid, it gets detached from this enzyme complex. So you are understanding what I am saying? Enzyme is not separating at each step, Okay, so acknowledge if you are understanding what I am saying. Can you guys hear me?
Yes, sir.
Yeah. So you are understanding what I am saying.
Yes, sir.
What I am saying is, whenever any reaction happen, substrate, enzyme and product, right? So substrate combines with the enzyme and it forms product. But on that product, no longer the enzyme is combined, right? In each step, enzyme detaches and then product is formed. But in fatty acid synthesis, the whole reaction keeps happening on the fatty acid synthase enzyme itself. So when the full fatty acid has formed, then only fatty acid synthase enzyme detaches. Okay, so here why that is important? Whenever the next step, the product will form from one step, the name will come as acyl malonyl enzyme because the enzyme is still attached to these substrates. That's why the name will keep coming enzyme. Normally, product name will not contain enzyme. So that's what I'm trying to tell you. Okay. So in first step of the fatty acid synthesis complex, acetyl transacylase and malonyl transacylase helps in bringing together the acetyl coenzyme A, one molecule of acetyl coenzyme A and one molecule of malonyl coenzyme A, forming acetyl malonyl enzyme. In next step, ketoacyl synthase will form ketoacyl enzyme. Okay. After ketoacyl synthase, the enzyme will come ketoacyl reductase. You have to find way how you are going to remember the steps. You don't have to remember the structure anywhere, but you have to remember the name of the substrate. You have to remember the name of the enzyme. You have to remember the name of the product. After ketoacyl synthase, the next enzyme is ketoacyl reductase. It is forming hydroxyacyl enzyme. After reductase, hydratase enzyme will come and form unsaturated acyl enzyme. Then enoyl reductase. So ketoacyl reductase, then hydratase, and then another reductase. It will form acyl enzyme. So here actually chain has formed. One small chain of fatty acid has formed. And this step, starting from the step 2, from here, basically, from here, so 1, 2, 3, 4. These 4 steps will keep repeating for the next 7 times. Okay, total 7 times it will repeat. Then total chain of the palmitic acid will form. And once total chain of palmitic acid is formed, with the help of thioesterase enzyme, fatty acid synthase complex will separate from this palmitic acid chain. Okay. Now another thing you have to remember is, like, very important cofactor required for this fatty acid biosynthesis is NADPH. So from where this NADPH is coming and where this NADPH is getting utilized. So NADPH get utilized in 2 steps. But the good thing is both the step name is reductase. So ketoacyl reductase enzyme uses NADPH as substrate and enoyl reductase uses NADPH as substrate. And it forms NADP. So this NADPH comes from, this is also important question, NADPH comes from 3 different pathways or enzymes. From where all it is from? Pentose phosphate pathway. This you must have read in carbohydrate metabolism. Next, it comes from isocitrate dehydrogenase. Third is malic enzyme. Okay, so from these 3 reactions, NADPH generation happen, and that NADPH is utilized in the fatty acid biosynthesis. So in one time you cannot remember or you cannot understand also, but you have to go back and read from book again and again. You have to try find your own way how you are going to remember these steps and these enzyme names. So this already discussed, no need of, I already told like this will be repeated 6 more times, so total 7 times, starting from the reaction number 2 of fatty acid synthase complex till the reaction number 5, it will be repeated total 7 times. Thioesterase, basically at the end when the full chain has formed, it will detach the fatty acid synthase complex. Okay, and then what will happen to that fatty acid synthesis? Like fatty acid synthesis happen, right? But only few, a small quantity of fatty acid remains in free form in the body. Rest all it forms other products. So what are those other products? So basically fatty acid can combine with glycerol. We already saw in lipid chemistry. It can combine with glycerol and form monoacyl, diacyl and triacyl glycerol. Or that palmitic acid can go chain elongation and it can form further longer chain fatty acids, right? Or it can go desaturation. Palmitic acid is a saturated, right, fatty acid. So it can go desaturated and form unsaturated fatty acids. Okay, or it can combine with cholesterol and form cholesterol ester. So these are basically the fate of fatty acid, you can say. This does not have much significance. Why this is important? Because it is specific for the C8, C10 and C12 acyl residue found in milk lipids. So in milk lipid, basically the fatty acid of chain length 8, 10 and 12 also comes. So for that there is separate thioesterase, which basically separates the fatty acid synthase after the few cycles only. And now acetyl coenzyme A, which was used as primer, formed the carbon number 15 and 16. Not much significant for you. Now I told you for even chain you need only acetyl coenzyme A. Okay, but for odd chain fatty acid, because odd chain will not contain the even number, right? It will contain one extra fatty acid. So for that the primer required is propionyl coenzyme A. It contains three. It contains 3 carbon, right? That is odd number. So this propionyl coenzyme A acts as a primer for odd chain fatty acid synthesis, and odd chain are mainly found in ruminant fat and milk. Ruminant fat is basically animals, you know that cow and all those animals are ruminant fat, contains ruminant fat. So now the balance sheet, what are the substrate involved in fatty acid synthesis and what are the product form? So this is what acetyl coenzyme, right? 1 2 carbon na? So this is acetyl coenzyme A. 7COH, because the first reaction of the fatty acid synthesis, total fatty acid synthesis, is by which enzyme? Acetyl coenzyme carboxylase. There we saw that bicarbonate is required, carboxylation of biotin happened, that carboxyl group gets transferred to the acetyl coenzyme and then it forms malonyl coenzyme. So malonyl coenzyme, this is acetyl coenzyme and this is malonyl coenzyme. So total 7 malonyl coenzyme and one primer acetyl coenzyme will be utilized, and it will again need 14 NADPH because 7 times total cycle is happening, and each cycle in each you saw that in each cycle 2 NADPH is required for 2 reductase enzyme and 14 H plus, and ultimately it will form palmitic acid. 14, 15, 16 carbon palmitic acid, 7 carbon dioxide will be released, and then coenzyme A will be separated and ATP will form. And where this 7 carbon dioxide is released, you will see here in this step. So this carbon dioxide is released in second step. Okay, this balance sheet you have to, if suppose in exam, I don't know whether this comes or not, it's a little complex, but if fatty acid synthesis comes, you have to draw the flow chart of the pathway. No need to draw the structure, write names of the substrate, product and enzymes, and at the end you have to write balance sheet, this thing. So this also you don't have to write in this form. You can write acetyl coenzyme A plus 7 malonyl coenzyme A plus 14 NADPH. This also is not compulsory. Then you can write palmitic acid plus 7 carbon dioxide plus 6 water and coenzyme A NADP. Now, what are the sources of NADPH for lipogenesis? Fatty acid biosynthesis is also known as lipogenesis, synthesis of lipid, right? So, first I told pentose phosphate pathway. So, where all it happens? In liver, adipose tissue, lactating mammary gland, and most of the fatty acid biosynthesis happens in liver and adipose tissue. And this both pentose phosphate pathway, if you remember, already in carbohydrate must be covered. This is also happens in cytosol, and this fatty acid biosynthesis also happens in cytosol. So for this NADPH, which is formed in pentose phosphate pathway, it is very easy to get incorporated into the fatty acid biosynthesis because both the cycles are happening in cytosol only. Then malic enzyme, the reaction we will see in the next step. Also here also you see malate plus NADP plus, it forms these reactions. You must be knowing that from malate pyruvate forms CO2 plus NADPH. Now cytosolic isocitrate dehydrogenase. Now isocitrate dehydrogenase is there in mitochondria also. You must have seen the reaction for mitochondrial isocitrate dehydrogenase in carbohydrate chemistry. Isocitrate plus NADP, it forms alpha ketoglutarate and NADPH. This is a malic enzyme reaction, malate, NADP forming NADPH. Just remember this. Okay, what are the sources of NADPH which is required fatty acid biosynthesis? First is pentose phosphate pathway, second is malic enzyme, third is isocitrate dehydrogenase. Everything is written in your book. Now, what is the source of acetyl coenzyme? Acetyl coenzyme is the most important, right? This is a building block basically of the fatty acid. So it comes mainly from glucose. So from glucose, pyruvate will form. This reaction, I am sure you all must be knowing, carbohydrate metabolism is over. From glucose, pyruvate will form, and this pyruvate ultimately what happens? It forms acetyl coenzyme in the mitochondria, if you remember the reaction. But this acetyl coenzyme which is forming in mitochondria, this is needed in cytosol for fatty acid biosynthesis, right? But this acetyl coenzyme directly cannot diffuse out of the mitochondria for fatty acid biosynthesis. So it first forms some other product. Ultimately, that product goes out of the mitochondria, and in the cytosol, that product again gets converted to acetyl coenzyme, and then it gets utilized. So remember, acetyl coenzyme is coming from the pyruvate and pyruvate is coming from glucose. Okay, but this acetyl coenzyme is forming in the mitochondria. This is needed in cytosol for fatty acid biosynthesis, but it cannot diffuse out on its own. So how it will diffuse out? Next step we'll see that. Acetyl coenzyme will combine with oxaloacetate in the mitochondria. It will form citrate. Okay, in the mitochondria this is happening. Now there is transporter, tricarboxylate transporter. If you don't remember the name, it's okay. Remember that there is transporter which transport out the citrate. Okay, now in the cytosol, ATP citrate lyase is present, this enzyme. Okay, and this enzyme will convert citrate to acetyl coenzyme. What was happening inside the mitochondria? Acetyl coenzyme A plus oxaloacetate was forming citrate. This citrate went out from the mitochondria into the cytosol with the help of a transporter. Just remember that. And there is a presence of an enzyme known as ATP citrate in the cytosol, which is converting citrate back to acetyl coenzyme A. Okay, that's it. It's easy. If you two three times if you read this, you'll be able to understand. Now this ATP citrate lyase increases in well-fed state. What is the meaning of well-fed state? It means there is no deficiency or a decrease in glucose in the body. Glucose is there, glycogen storage is sufficient. Then whatever excess glucose is present in the body, that when it gets converted to acetyl coenzyme, and that is not needed for energy production or other purpose, it will get converted to fatty acid. That's why in well-fed state, this enzyme activity will increase so that more and more acetyl coenzyme present in mitochondria ultimately gets converted to acetyl coenzyme outside the mitochondria in the cytosol so that it can get incorporated into the fatty acid. And this oxaloacetate which is formed in the cytosol will get converted to malate, and ultimately in with the help of malic enzyme it will form NADPH, right? And in the pyruvate, in the carbohydrate metabolism, you have read that from pyruvate acetyl coenzyme may form. Okay, so sources of acetyl coenzyme its fate. Mainly acetyl coenzyme comes from pyruvate. It can also come from ketogenic amino acid. For you, protein metabolism is also over, I think, and amino acid metabolism. So you must be knowing that there are some amino acids like ketogenic, some are glucogenic, and all like that, right? So from the ketogenic amino acid also acetyl coenzyme can come. Mainly it comes from pyruvate, which comes from glucose, but some acetyl coenzyme can come from amino acid. Some can come from the oxidation of fatty acid. Fatty acid can get oxidized, right? Like how fatty acid is forming, fatty acid can get broken down also. So once fatty acid breaks down, it will form acetyl coenzyme. And this acetyl coenzyme can be utilized in different various ways. What we are discussing here is acetyl coenzyme forming fatty acid. Okay, it can form steroids also, it can form ketone bodies also. Ketone body also we will read in lipid only. I'll take class. And then you know that if fatty acid, acetyl coenzyme, most important function is in TCA cycle, right? So that you already know. So this is the source, this is the fate. So from pyruvate, from ketogenic amino acid, from fatty acid, acetylcholine is coming, acetyl, and then it gets converted into, or it forms fatty acid, steroid, ketone bodies, and TCA cycle. Now that fatty acid can form TG, triacylglycerol, right, while combining with glycerol, or its chain elongation can happen. For chain elongation, a small organelle which is present in the cell is microsome, right? So there microsomal elongate system is present, that helps in the elongation of the fatty acid chain further. Like palmitic acid is 16, but if suppose you have to make some other fatty acid of longer chain, then you need some other enzyme for elongating this fatty acid chain. So there the function of role of microsomal elongate system comes. For both saturated and unsaturated fatty acid, this microsomal elongate system help in the elongation. And here manual coenzyme and ADPH will be required for the elongation also. Fate of fatty acid. So most of the fatty acid either synthesized in the body or you can ingest also, right? So what happens? There are two main fate. Either it will get incorporated into triacylglycerol for the storage of metabolic energy. So that's why if you take more and more glucose, more and more carbohydrate, more and more pyruvate will form. Ultimately, more and more acetyl coenzyme F4, and from there more and more fatty acid will form, and from there more and more triglyceride will form. And if more and more triglyceride keep forming, that will get keep storing in the liver, leading to fatty liver and all. So either it can get stored for metabolic energy or incorporated into the phospholipid. So ultimately this TG can form phospholipid also, right? We saw that glycerol, fatty acid, and along with that phosphate group plus nitrogenous bases can come and combine and form phospholipid. Now regulation. So it can have nutritional regulation, right? Excess carbohydrate in the lipogenesis will increase, right? Glucose, pyruvate, lactate, and acetylcholine will help in the formation of more fat. Now how it will decrease? This fatty acid synthesis can be decreased by restricted calorie intake. If you like less calorie food, then the fatty acid biosynthesis decreases. High fat diet. Okay. If you're taking high fat diet, the formation of fatty acid inside body will decrease. Insulin deficiency. I told you that insulin is required for the formation of the fatty acid. So if there is insulin deficiency, especially in type 1 diabetes, now there is inverse relationship between hepatic lipogenesis and serum free fatty acid. It's simple. If hepatic lipogenesis is happening, it means what? This free fatty acid is getting utilized in forming the TG and all, triglyceride and all in the liver, right? So the serum free fatty acid will decrease if more and more lipogenesis is happening in the liver. Sucrose feeding increases lipogenesis. Why? Ultimately, sucrose contains fructose, and you must have read that PFK, na, phosphofructokinase, its role in the glycolysis and all, glucose and all, right? So that checkpoint is bypassed by fructose. For fructose metabolism, this PFK does not have role, so it is not able to control the formation of pyruvate. So pyruvate forms from glucose also, pyruvate will form from the fructose also, right? From the fructose, ultimately pyruvate. But there is no controlling step by the PFK enzyme. So that's why more and more pyruvate can form if you take more and more fructose, and that will lead to more fatty acid synthesis. Now regulation of lipogenesis in enzyme is over, so you must be knowing like how different types of enzyme regulation happens, right? So in short term regulation, allosteric and covalent modification happen. So I told you the rate limiting enzyme and the most important enzyme of the fatty acid biosynthesis is acetylcoenzyme carboxylase. This is the allosteric enzyme. So allosteric modification of acetylcoenzyme happen, or there can be covalent modification happen also, and depending on whether this acetylcoenzyme carboxylase is required or not. So when it will be required more? When fatty acid biosynthesis will be required more. If so, depending on the requirement of the acetylcoenzyme carboxylase for fatty acid biosynthesis, its allosteric and covalent modification will happen, making it either active or inactive. But this will be like short term. But if long term regulation has to happen, the gene expression of the enzyme itself changes. So the enzymes involved in the fatty acid biosynthesis, their gene expression will be controlled. So the enzyme itself will not form. Either it will not form or it will form more. More and more transcription will happen and more enzymes will form in the long term. Citrate, it is activator. So it increases in well-fed state. So if well-fed state, more and more fatty acid biosynthesis will happen because you are taking more glucose, more. So that is ultimately going to form more fatty acid. citrate converts the enzyme from an inactive dimer to an active polymeric form. Okay, so if active polymeric form is there, then the fatty acid synthase enzyme will be more active. Now this fatty acid synthase enzyme is inactivated by long chain acyl coenzyme. So acyl coenzyme is nothing, basically it is fatty acid. So if long chain fatty acid keeps forming, then the enzyme by negative feedback loop, it will get inactivated. Then activation, inactivation, you must have read by phosphorylation in enzyme. So by if this enzyme gets phosphorylated, this lipogenesis is inactivated basically. Now another thing is acyl coenzyme is nothing but fatty acid. Fatty acid, you have to understand. Fatty acyl coenzyme. Acyl, the meaning itself is fatty acid basically. Inhibits mitochondrial. If more and more fatty acid is already forming, then it will inhibit the transporter. So what is the significance of that? So from mitochondrial transporter, if fatty acid has formed a lot in cytosol and it will inhibit the mitochondrial transporter, so the further coming out of acetyl coenzyme from mitochondria to cytosol will not happen, because now it no longer acetyl coenzyme is needed in the cytosol because already so much fatty acid is formed. So that's why this fatty acyl coenzyme, which is basically the newly formed fatty acids, inhibit mitochondrial transporter, inhibiting the coming out of acetyl coenzyme from the mitochondria to cytoplasm. So this is what prevent the egress of citrate. This citrate only breaks down and form acetyl coenzyme. Acetyl coenzyme carboxylase is also regulated by glucagon, epinephrine, and insulin. So basically, insulin increases the activity of acetyl coenzyme, and then glucagon and epinephrine will have opposite action. This you know. Next is role of pyruvate dehydrogenase. See here. So if more and more acetyl coenzyme is there, so this ratio will be more acetyl coenzyme by coenzyme ratio. More and more NADPH is there, so NADPH by NADP ratio will be more. More ATP, less ADP. So if this ratio is more, it activates plus plus plus. It activates PDS kinase, right? And this leads to inactive PDH. So basically, if more and more acetyl coenzyme has already formed, so ultimately it will lead to inactivation of PDH, and this PDF will help in the reduction in formation of acetylcoenzyme. As how acetylcoenzyme is forming? It is forming from pyruvate to acetylcoenzyme with the help of pyruvate dehydrogenase. But if already so much more of acetylcoenzyme form, then by negative feedback loop it will inhibit PDH. Simple, okay? More acetylcoenzyme will inhibit PDH. Role of insulin, this is important. It activates acetyl coenzyme carboxylase. I told na, like, insulin promotes formation of fatty acid, increases the transport of glucose into the cell. It increases the role of insulin. I am telling, increases the availability of pyruvate and glycerol, and it converts inactive form of PDH to active form of PDS, because inactive form of PDH will not form acetyl coenzyme, so it converts it to active form, and then more acetyl coenzyme will form, more fatty acid synthesis will happen, and it inhibits lipolysis. So insulin, what happens? Ultimately, role of insulin is to decrease glucose in the circulation. Okay. For that, what it does? It takes up those glucose inside the cell and help in the formation of fatty acid. So ultimately, it is taking glucose from the circulation and forming fatty acid. That's it. And it inhibits lipolysis from the cell also, so that this free fatty acid does not come out into the circulation. Long-term regulation, I already told it, these are adaptive enzymes, and with the help of modified gene expression, the synthesis of fatty acid synthase complex and acetylcoenzymic carboxylase can either decrease or increase. I already told in well-fed state it increases. Okay, it is decreased starvation, because that time fatty acid breakdown will happen, lipolysis will happen to give energy, right? So that time more and more fatty acid cannot be utilized. Another reason why it cannot be utilized, because in the first step of the reaction you have seen that fatty acid uses ATP, right, in the formation of what, malonyl coenzyme from acetyl coenzyme. So that time more and more ATP is required for the energy. It cannot be utilized by the fatty acid biosynthesis. Insulin agonizes means insulin promotes fatty acid biosynthesis. Glucagon inhibits fatty acid synthesis. And polyunsaturated fatty acids inhibits the expression of enzymes of lipogenesis and glycolysis. This is over. Now we read about different types of lipids, right? Compound lipid and all we saw. Compound lipid, complex lipid and all. But sometimes those lipids can accumulate in excess in the body and can cause inborn error of metabolism. Those like they, because of the accumulation, excess accumulation of different types of compound and complex lipid, especially phospholipids and gangliosides, it causes inborn error of metabolism of lipid in the infants, leading to different types of diseases. I don't think it comes in your exam, but two, three are important. Just know the name. Gaucher disease, it is very common, involvement of metabolism in children. It happens because of accumulation of glucosyl ceramide. We read about ceramide, right? Non-phosphorylated lipids. You must have read, na? So glucosyl ceramide, if it gets accumulated, it will form. It will cause Gaucher's disease. Why that is happening? Because the enzyme deficiency is beta glucosidase. So if this enzyme was there, they would have broken this down, glucosyl ceramide. Now the accumulation of galactosyl ceramide, because of the deficiency of beta galactoside, Krabbe's disease happen. Tay-Sachs disease happen because of, all these are in your book. I am telling you just what, which all are important, more important. Tay-Sachs disease are hexosaminidase A. This is because of the accumulation of ganglioside. And Niemann-Pick disease, deficiency of sphingomyelinase. Sphingomyelin, you have read, right? So sphingomyelin, if it gets accumulated because of the deficiency of sphingomyelinase, Niemann-Pick disease happens. And mostly the symptoms will be very common, like the child, infant will have mental retardation, muscular weakness, then liver and spleen will be enlarged, skeletal deformity, skin lesions, so kidney failure, so many problem happens. So you can go and see these like important only. I don't think they will ask you such things in your exam, but anyway, you should know. Now next class will be on oxidation. So right now we saw fatty acid biosynthesis, how fatty acid is forming. Next you will see fatty acid oxidation, how fatty acid is breaking down and to really give energy, right? So what is the difference? Right now itself we will see. We saw that biosynthesis happens in cytoplasm, but the oxidation pathway happens in mitochondria. Intermediate is linked to the ACP. Okay, those like those malonyl coenzyme, acetylcholine, those are linked to acyl carrier protein. Here, the oxidation is in intermediates are coenzyme A derivative. Sequential unit, two carbon unit from the acetyl coenzyme A. It keeps forming the acetyl coenzyme A, but in biosynthesis, two carbon unit gets attached, but it comes from malonyl coenzyme A. An enzyme, here all the enzymes are independent. Here we saw that in biosynthesis the 7 enzyme multi enzyme complex was there, fatty acid synthesis, right? Coenzyme, very important coenzyme for biosynthesis is NADPH. Here for oxidation you need NAD plus and FAD. We will see this difference, you will understand when I take the fatty acid oxidation class in the next. Okay, so that's it. You have to go back and read. No need to read a lot. What you have to read is where the fatty acid biosynthesis happen, what are the enzymes involved, what is the sequence of the reaction. No need to know the structure, just remember the names of the substrate, enzyme and product. Okay, what is the most important enzyme, rate limiting enzyme? Acetyl coenzyme A, carboxylase. What are the cofactors required? From where NADPH comes? Okay, and how the regulation happens. So important roles, like what is the role of insulin? It increases, glucagon decreases, PDH. If acetyl coenzyme A increases, PDH is inhibited. Okay, if you are in well-fed state, like body like is getting proper amount of glucose and all, then fatty acid biosynthesis increases. But if you are in starvation, fatty acid biosynthesis decreases. So important points you remember. Go and read and make notes. Okay. So that's it for today. I will stop the presentation. Thank you.
Thank you, sir.
Thank you, sir.